And thank you everyone for coming to this seminar at lunchtime. So fortunately, I decided to talk about sodium calcium channels, not glucose transporters, not to make you more hungry. It's certainly a great pleasure to be here today. So it's a shame that, I have been at Princeton for 7 years as graduate student and postdoc, and then I came back last year. But it's really the second time that I visit Rutgers. The first time I came here was in January when I came here to discuss with Wei about some potential collaboration on electron tomography. Okay, so as we just told you, so I set up my lab in Tsinghua in 2007. Actually, that was not the point that I started working on membrane proteins. I stayed in Yigong's lab for PhD study. During that period, I was focusing on apoptosis. And then the reason I didn't leave Princeton was, Yigong asked me to start on this new direction that was the structure determination of intramembrane proteolysis. So basically we solved one crystal structure named GlypG, which is a membrane embedded protease. That's when I was absorbed to this field. And then when I started my own lab, of course, you want to do something different from your postdoc research. So I started, working on the so-called transport proteins. So by transport proteins, I actually refer to two major classes, channels and transporters, as summarized here. So our cell is surrounded by lipid bilayers. Well, these membranes define the cell, protect the inner contact of the cell or organelle from the environment. This lipid bilayer also represents a barrier for free exchange of chemicals or ions, or information. So basically, and there are several ways for the chemicals or even larger like proteins to move across this membrane. So for small molecules such as oxygen or glycerol water molecules, they can penetrate the lipid bilayer through simple diffusion. But for ions and larger chemicals such as amino acids or sugars or even peptides, there's no way for them to penetrate through diffusion. Basically, there are several different approaches. Like you can do that through exocytosis or endocytosis, right? That's called the group or vesicular translocation. But throughout probably billion years of evolution, different types of proteins have been evolved to facilitate this process. So these proteins can be classified to channels or transporters. And you may ask, what's the difference between channels and transporters? So different people in the field may give you a different definition. But in my mind, actually, these two classes of proteins are distinguished based on their mechanism. Basically, shown here is a simplified diagram of channel. So once the gate, so the channel is just not a pore, okay? It's it's tightly regulated. It contains gates to insulate this kind of permeation path from the environment. But for a channel, once the gates are open, the permeation path is completely accessible to both sides of the membrane. Therefore, a channel can only mediate, the movement of its substrate down its concentration gradient or its electrochemical potential. So basically, it cannot catalyze the uphill translocation of substrate. In contrast, a transporter contains at least two gates. So once the gate is, at any given moment, the substrate binding site is insulated from at least one side of the membrane, meaning that at any given moment, at least one gate is closed, all right? So we call these two gates intracellular gate and extracellular gate. So basically the model is a transporter may contain one or multiple substrate binding sites, but at any given moment, this binding site can only be exposed to either side of the membrane. This is called the alternating access. So, through this mechanism, basically a transporter has the ability to catalyze the uphill translocation of a substrate. This is the ability that transporter has, but a channel does not have, all right? So that's the difference. But on the other hand, it's not necessary for transporter, to be an active transporter. So transporters also, contain the so-called uniporters, such as the glucose transporter. Basically, it only catalyzes, the kind of diffusion of a substrate through this transporter. basically it's down its concentration gradient. But again, it undergoes the so-called alternating access. So it's still classified as a transporter or carrier, but not a channel. Okay, hopefully at this moment I don't confuse you. All right, in MATLAB, so this is kind of a basic introduction to transporters and channels. So in MATLAB, we have been particularly interested in two types of transport proteins. The channels exemplified by the voltage-gated sodium-calcium channels, which I will elaborate in details today. And the other type is the sugar transporters, exemplified by the glucose transporters in human. So basically, our primary goal, of course, is to review their architecture, their structure, right? That's kind of the primary, also the superficial goal. And by solving the structures, we'd like to understand their mechanism, how they selected the substrate, what's the molecular basis of the, substrate selection, and what's the basis for, the energy coupling, etc. And by working with different types of transporters and channels, we hope to elucidate the chemical and physical principle of membrane transport in general. So, in the past several years, I'm very lucky, I have very talented graduate students and postdocs who are able to solve the structures of the human GLUT1 and its close cousin GLUT3 in 3 distinct conformations. So basically, this three structures give us an overall picture of what, how these transporters specifically select glucose, and how they complete the so-called alternating access cycle. So if you're interested, I actually have an iBiology seminar online, so about glucose transporters. So today I will just switch to the second part of, the second focus of my lab. I have to say that we started working on glucose transporters and voltage-gated sodium channels in 2007. All right, so all these projects, although it seems like we solved them just within 2 years, but it has been like 10-year long, a decade-long project. So the second part is on the molecular basis of the initiation of electrical signaling. So for in multicellular organisms, there are multiple ways for signal transduction, but for this instant signal transduction, electrical signaling plays an important role. For instance, so here's a kind of a bonfire. If you just use your hand to touch, okay, a flame, all right, so instantly you will just withdraw, right, because it's kind of it's burning. So how can you sense this burning? It's by electric signal. If it's by kind of a chemical diffusion, probably it takes like several days for your brain to feel this kind of this pain, and then takes another several days for you to move back. So at that time, you no longer have your fingers, all right? Okay, so electric signal is important. Then how does our body really, kind of perform this electrical signaling? So several types of channels called voltage-gated ion channels that are really underlie this electric signaling. So among different channels, the voltage-gated sodium channels, so I will refer that as Navh channel from now on. So sodium channels, actually Navh channels are responsible for the initiation and propagation of action potential. As shown here, this is a simplified diagram. I believe most of you have learned that from textbook. So basically, in excitable systems such as neuron systems and muscles, so the membranes are polarized, meaning that the ions are distributed unevenly. So due to the asymmetric distribution of ions such as sodium ions and potassium ions, there is a electric field across the membrane, which we call as the membrane potential. Okay, so in this cells, if it's at resting state, the membrane potential is negative, meaning that it's negative inside. All right, but this membrane potential fluctuates when it rises to certain threshold, as shown here. At resting state, it's about like -70 millivolts. But when it rises to about like -55 millivolts, the voltage-gated sodium channels are activated. So that would lead to a rapid influx of sodium ions, resulting in this rapid rising phase of the membrane, potential. So we call it the basically up phase, or this actually is the initiation of the action potential. Meanwhile, the sodium channels quickly inactivate, and the voltage-gated potassium channels also activate to lead to the efflux of the potassium ions to reset the membrane potential back to its resting state. So this is kind of a complete action potential. So this is really a fundamental physiological process, right? So due to a fundamental significance, defects or misregulation of sodium channels are associated with a broad spectrum of diseases or disorders, such as the pain syndrome, cardiac arrhythmias, and also epileptic seizures. So it's not surprising that this voltage-gated ion channels, not only the sodium channels, but also calcium channels and potassium channels represent major drug targets, all right, as shown here. And as a structural biologist, I'm particularly interested in two fundamental questions. Actually, I already mentioned those. First, what is the molecular basis underlying ion selection? As shown here, why a Navh channel just strictly permeates sodium ions, but not potassium or calcium. Remember, calcium, sodium actually share very similar radius, okay? But sodium channels will block the penetration of calcium, vice versa. And a more intriguing question is, how does this channel sense the fluctuation of membrane potential and translate that into the opening or closure of the central pore? So this is called the voltage-dependent gating. By this gating mechanism, I mean both activation and inactivation. So these are the central questions that we'd like to address through structural study of different types of Navh channels. And our understanding of these channels actually came from the pioneering work of Ron McKinnon's lab. On the left is Nobel-winning structure of KCSA, but it's just a kind of half of the pore domain. Basically, shown here is the diagonal protomers of the pore domain of KCSA, which only contains pore domain. Okay, it's a bacterial homolog, as shown here. This inner helix bundle and outer helix bundle. So basically, they constitute this selective filter that represents the first barrier for ion selection. And then a few years later, so they solved the structure of the first voltage-gated potassium channel. As you can see, this is a side view, okay? So this is a side view of the pore domain. And on the right is basically the extracellular view of voltage-gated potassium channel. So in the center is basically the pore domain, almost identical to the one shown on the left. And surrounding this pore domain, there are four kind of loosely attached voltage sensors, also known as the voltage sensing domains, VSDs. And sequential analysis of all the voltage-gated ion channels, not just KV channels, but also Navh and Cav channels, shown that all these channels share similar architecture. Basically, they are either homotetramer or homodimer, or like a single chain that are folded to 4 repeats. So basically, these channels would have either perfect 4-fold symmetry or pseudo 4-fold symmetry. Okay, so each subunit or each repeat, basically there are 6 transmembrane helices named S1 to S6, and among these 6 transmembrane helices, the 4 sets of S5 and S6 enclose the central pore, as shown up here. Okay, so that's kind of 4 sets of S5 and S6. And then, with each repeat, the 4 preceding helices, S1 to S4, fold an independent voltage sensor. And among these 4 helices, you can see, S4 is particularly interesting because it carries this repetitively occurring positive charge residues, arginine or lysine. And so based on this sequence analysis, a model was proposed to explain, how these channels are, kind of respond to the change of the membrane potential. According to this model, so basically S4 would use this, positive charges to sense the fluctuation of membrane potential. At the resting state, when the intracellular side is more negative, so S4 is attracted to be on the intracellular side, all right? So during, the depolarization, S4 is pushed outward, and this conformational change of S4 is connected to the pore opening on the intracellular side through a linker, S4, S5 linker. So this is a very neat model. On the other hand, if you'd like to design a voltage-gated ion channel, probably you need 3 elements, right? So you would need ion selective filter, right? I mean, either it's for cation or anion or sodium or potassium or calcium. So you need a selective filter. And then you need a gate on the other side that would prevent the leak of any ions. And then you need this voltage sensor. And this sensor has to, couple its conformation change with the opening or closure of this intracellular gate. So this model is very neat, but if you think about it, how can the possibly charged segment move freely across this hydrophobic lipid environment, right? So there will be, energy barrier to prevent the conformational change of S4. And sequence analysis of, other segments in VSDs identified two conserved polar or negative charge residues on the opposing S2 segment. So these are named ion 1 and ion 2. And so the model was that, so when S4 undergoes this translational or other types of movement, so these positive charge residues, which are named the gating charges, would form a sequential interaction with the ion 1 and ion 2 to lower the energy barrier. So these are all very neat models, but whether that's the case or not, so what's the detailed mechanism underlying this electromechanical coupling remains to be investigated. Oh, by the way, so also it was the pioneering work from Ron McKinnon's lab also identified the so-called charge transfer center that comprises both this negative charge residue and another one that's now shown on the S3 segment, as well as including hydrophobic residue on S2 to be the charge transfer center that may facilitate the movement of S4. Of these gating charge residues. And shown here is a summary of the members in the voltage gated ion channel superfamily. As shown here, CV and NAV, CAV and Navh channels actually are closely related to each other. And they are unique in that they consist of one single polypeptide chain that folds to 4 repeats. And then, on the other hand, potassium channel actually has more subtypes, mostly are homotetramers. But in the case of K2P, the 2-pore channel, so it's a dimer. Basically, they have like 2-fold symmetry and 4-fold pseudo-symmetry. And then the very famous trip channels also belong to this family, although they may not be voltage gated. Some of them are temperature sensitive or mechanical sensing or even ligand gated. But because they share identical folds with VGSE proteins, so they are also classified to the VGSE superfamily. And as I told you, on the sodium channels and calcium channels from eukaryotes are very unique in that they only contain one single polypeptide chain that folds to 4 similar but non-identical repeats. So this feature made them basically the most challenging targets for structure determination. As you can imagine, they are huge, right? So it's difficult to generate a recombinant express protein. And also, they are subjected to heavy post-translational modifications, such as glycosylation or phosphorylation. So it's almost impractical to crystallize these proteins. I mean, we have tried for years, all right? So for several years, but never succeeded. So to my knowledge, no one, I might be wrong because people don't publish unsuccessful data, right? So I really don't know whether anybody has ever successfully crystallized these proteins. Fortunately, on Earth, apart from eukaryotes, there are bacteria, right? And sometimes these weird bacteria may contain weird proteins. We don't know their function, but sequence analysis would tell you that, oh, they belong to, Navh family or CAV family. And in the case of voltage-gated sodium channels, there are indeed such bacteria homologs. So in 2001, Dave Clampham's lab in Harvard University identified a family of bacteria proteins. They named these proteins as NavPas for sodium selective channel of bacteria. So basically, these proteins actually share sequence similarity more to CAV channels. But their electrophysiological analysis identified to be sodium selective. The best thing about these NavPas proteins is that they are homotetramers, unlike their eukaryotic, not cousins. I don't know, maybe they're kind of great-grandson or something. Anyway, so they are homotetramers. So each protemer is small. It contains less than 300 amino acids. So, and when we saw this, very kind of thick band, I was surprised that no one has ever solved this structure in 2007, although it was identified in 2001. So in my own lab, I persuaded a junior student to start working on this one, and he could never ever reproduce this band. All right, so we went back to yeah, they are over there. You can see this band. So we went back to read the the paper in details. Turns out this is Western blot. It's not Kumasi blue staining. So that's why you have to read the paper in detail, all right? to cut the long story short, so we could not produce large amount of NavPas, but anyway, this bacterial homolog. So after a couple of years, we were able to identify another bacterial homolog, which is homolog of NavPas from a marine bacteria species. So I even forget the name of this bacteria. So it's basically species names RH, all right? So we solved the structure of NavRH. As you can see here, it contains this central pore domain surrounded by 4 voltage sensors. But I have to tell you, this was not the first structure of NavPas, it was the second one. And a year ahead of our publication. So basically, Nieng and Bill Cattrall from University of Washington published the structure of another bacteria, sodium channel. They named it NavAB, also after the species name. And surprisingly, when we superimpose our structure to theirs, ours is colored green and theirs is gray. You can immediately tell that these two proteins adopt different conformations, right? Their VSDs undergo this kind of rotation, and this gave us the very unique opportunity to examine, the ion selection or the gating charge transfer. To cut the long story short, we collaborated with the computational biologist in Tsinghua, Dr. Haipeng Gong, to perform MD simulation to study the ion selectivity. But I will not go into details because in retrospect, whatever information we got from here cannot be translated to the eukaryotic proteins, although they are sodium selective, but the mechanism and the molecular details are totally different. And then, for the gating charge transfer, remember, this is ion 1 and ion 2, and this is the kind of occluding residue IS2. The gating charge transfer is in a way for these gating charges to go across this occluding hydrophobic residue. So structural comparison of NavRH and NavAB vividly reveal the process of this gating charge transfer. So that's some information derived from structural comparison of 2 different proteins. But that's something fun to look at. But because it's of different proteins, so people can always argue with this artifact, right? So actually, we solved the structure in 2011, and took us 4 years try to verify this process without success. I have to say, sometimes structure determination actually represents the easier step. All right, okay, I will just stop here for the bacterial homologs. Even there have been 2 structures of bacterial homologs, and then 2 years ago, a third one was published, all right? So it's indeed difficult to solve such structures. Even with this structure information, there are several questions that are unique to the bacteria Navh channels, especially for human Navh channels. First, as told you, as I mentioned several times, that these channels are made of a single chain. So in a sense, the arrangement of 4 repeats was unknown, right? Theoretically, there are 6 ways to arrange the 4 repeats. So even this simple question remained unknown for a long time. And then because of, the sequence variation of the 4 repeats, the eukaryotic Navh channel actually has 4 distinct residues that determine, 1 on each repeat, that together determine the sodium selectivity. So DEKA, this is unique only to eukaryotic Navh channels, because in bacteria channels and also potassium channels, it's homotetramer, right? So it's identical. And even in the eukaryotic calcium channels, it's the same. 4 glutamate residues define calcium selection. So it's AEEEE. And only in eukaryotic sodium channel is DEKA for completely different residues that define sodium selectivity. So how does that happen remained completely unknown. By the way, we also did some modeling based on bacterial homolog, and in retrospect, it was completely wrong. So sometimes you have to, take caution when interpreting, kind of models. Okay, and another question is, it has been characterized that these 4 repeats actually are not activated simultaneously. For all the homologs or subtypes characterized always follow this sequential, like repeat 3 activates first and followed by repeat 1, then repeat 2, and then repeat 4 is activated in the last. Actually, activation of repeat 4 immediately leads to inactivation. So this kind of complicated process. So what take-home message is these 4 repeats are activated asynchronously, okay? And then another fascinating mechanism is a fast inactivation. As I told you, upon activation of sodium channel, it immediately inactivates to ensure the repetitive occurrence of action potential, right? You have to shut it down immediately. So this is called the fast inactivation that occurs at a millisecond time scale. And you know, the sodium channel has been identified for almost half century, and there have been, you know, enormous volume of literature on it. So it has been suggested that a linker that connect repeat 3 and repeat 4. So I will refer to as 3-4 linker. Okay, so basically a sequence on the 3-4 linker that would perform this fast inactivation. So basically 3 hydrophobic residues on the 3-4 linker, IFM or LFM, leucine, methionine, and phenylalanine, highly hydrophobic residues that may constitute the so-called fast inactivation particle. So upon activation of repeat 4, so this fast inactivation particle will be repositioned to block the central intracellular gate. Okay, so this is called the ball and the chain model. But you know, this model has been challenged in the past 10 years, whether it's right or not. And last but not least, so sodium channels actually also represent the major target for naturally occurring toxins. You know, there are so many different types of toxins that target sodium immediately, like, you know, the toxins from the snake, scorpion, or coral snails, or the pufferfish. You know, basically the major target, primary target, sodium channels, and which also make the sodium channels perfect target for painkillers, all right? I mean, if you think about it. As a matter of fact, sodium channels are also the major target for the local anesthetic drugs. So, but unfortunately, so far, there's no structure available for any sodium channels in complex with any toxins, because these sites are not conserved in the bacterial homologs. So, you know, to review like the molecular basis for all these questions that I just mentioned really requires structural determination of a eukaryotic sodium channel. All right, so as I mentioned, and this is the second time I show this, sodium channels and the calcium channels are close homologs. So our primary focus was on sodium channels, but meanwhile, we are also interested in the CAV channels. So we identified a very important and interesting system to work on. That is the so-called excitation contraction coupling of muscles. As shown here, like our heart and our skeletal muscle can beat, can move because of the electric signal, and they control. This process is initiated by several different types of channels. Shown here is a simplified cartoon of the contraction of our skeletal muscle at different scales. So here is actually at the cellular level, and then on the molecular level. So basically, upon the arrival of the signal from motor neuron, the membrane of the muscle cells is depolarized, so which would initiate the membrane potential, right? So sodium channels are involved, conducting this propagation of action potential. And when action potential arrives at a specialized membrane structure known as the transverse tubule or T-tubule, which is really the invagination of the membranes, where another type of channels, the voltage-gated calcium channels reside. So basically, these CAV channels in skeletal muscle is CAV 1.1, okay? So CAV 1.1 activates upon membrane depolarization. And activation of CAV 1.1 leads to the activation of another downstream channel known as the ryanodine receptor, so RYR1. So RYR is actually the largest ion channel known of today, okay? And it's named as a receptor, but it is actually the high conductance calcium channel that is responsible for the rapid release of calcium from the intracellular calcium store, such as SR or ER, to the cytosol. So once again, sodium channels are responsible for the generation and propagation of action potential. And within T-tubule, calcium channels activate in response to the action potential. And then, I mean, owing to the direct physical interaction, the RYR on the SR, I mean, sarcoplasmic reticulum would be activated through the conformational change of CAV 1.1. And then the release of calcium would trigger a cascade of events that eventually leads to muscle contraction. It's also kind of very fascinating, but our major interests are in the 3 ion channels. So in the past, I'm pretty happy that because of the resolution revolution of Cryo-EM, we were able to solve the structures of all 3 types of channels at near atomic resolution and in multiple conformations as shown here. So we solved the structures of RYR1, the one in the skeletal muscle, and also RYR2, the one in the cardiac muscle, in multiple conformations as shown here. And then, you know, a year later, we solved the structure of CAV 1.1, the calcium channel in complex with its auxiliary subunits. So I'd like to draw your attention to their molecular weight. For crystallization, we always try to truncate the protein to make it as small and as compact as possible, right? But for EM, we must know that it's the bigger the better. So our first structure was RYR1, you know, which has molecular weight of 2.2 megadalton. And then like the structure of the CAV 1.1 complex was resolved a year later. This complex had molecular weight of about 400 kilodalton. So after this practice, you know, we gained a lot of experience and also learned a great deal of lessons. And eventually we were able to solve the structures of 2 eukaryotic sodium channels, which have molecular weight of about 200 kilodalton. All right. So now, I mean, you know, the structure, the resolution, reported resolution is about 3.8 angstrom or 4.0 angstrom. But for EM map, this resolution is very decent to allow, you know, for the assignment of the side chains. So with the structures, now, you know, we can address the questions that I asked earlier. All right, so first, let me show you, you know, some details about structure determination. All right, I mean, sodium channels, you know, I have been talking about sodium channels because they're so important. There are 9 subtypes of sodium channels named Navh 1.1 to Navh 1.9 in human, okay, as shown here. And different types of sodium channels have distinct tissue distribution and are responsible for different type of diseases because of their tissue distribution and distinct functions. And then, you know, of course, our primary goal was to solve the structure of a human sodium channel, which is more physiologically and pathologically relevant, right? But, you know, you have to make sure, yeah, you know, for structural biology, it's not what you want to do, but what you can do. So we have spent like several very talented graduate students have spent several years struggling with the expression of the human sodium channel without success. Eventually, a very talented graduate student, Huaizong, you know, came up with a great idea. He was thinking that, you know, I won't do screening. Basically, you know, our success with the structural determination of that bacteria homolog, NavRH, was that protein actually was the shortest homolog of all the bacteria sodium channels. Along this line of thinking, what if, you know, I look for the shortest homolog of a eukaryotic sodium channel? So that's what he did. And that's why, you know, our first protein is called NavPas. It's a homolog from the American cockroach. It's a short form of American cockroach, which is the shortest one among all the deposited sequences of eukaryotic Navh channels. So basically, you know, we're able to express the protein and already, you know, show you like the yield is still extremely low. We can only obtain 50 microgram of proteins out of 8 liters of kind of suspension mammalian culture. So this low yield make it impossible for crystallization, but still, I mean, give us sufficient amount of protein for cryo sample preparation. And then, you know, that was the first bottleneck. I mean, to generate, you know, kind of sufficient amount of well-behaved protein, right? But then, like, you know, we collect data, you know, the cryo data, and then here came, you know, the second bottleneck, because the protein is only 150 kilodalton. That's still relatively small. I mean, what's worse is that the protein is embedded in determined micelle. It does not have distinct feature out of the membrane region. Okay, so what you can see is just like some round disc. it's extremely difficult to do the alignment. So it took us like several months just to struggle with the data processing. And then my very talented senior member, Dr. Qiang Zhou, who has been in this business, I mean, in Cryo-EM business for more than 10 years. So it just occurred to him that, the particles cannot be well aligned probably because, I mean, during the conventional pipeline, like 2 steps were mixed. That is, the selection of good particles and the alignment of this, 3D classification of the good particles. What if I separate these 2 steps? So basically, simply set, K21 just to select the good particles and then assign the alignment parameters to each particle, and then which would serve as the reference value for the later on 3D classification. And then, that's just worked. So as you can see, out of, for the second step, out of 10 classes, 1 class just stands out. So with this, I mean, everything became so straightforward. And he actually has several other tricks that, has been applied in other projects with, small membrane proteins in my lab. So basically with that, we were able to solve the structure of NavPas to 3.8 angstrom. And I intentionally aligned, the eukaryotic sodium channel side by side with, the bacterial one. So you can immediately see the difference. Of course, I mean, this is homotetramer, and this is kind of just a single chain protein. And this eukaryotic Navh channel contains 2, intracellular domains, which were unresolved before, and also, above its pore domain, we observe a heavily glycosylated, loop region. So these loops are surprisingly rigid because of the disulfide bonds. Okay, so we call it the window, the dome above the, the kind of the selective filter, so which is highly glycosylated and also enriched of negative charge residues. So it may help attract the cations. All right, this is the overall structure of NavPas, the first structure of eukaryotic Navh channel whatsoever. And you know what that is? It's called electric eel. Okay. So how can it generate this electricity to hunt or defend? Because of sodium channels. So as a matter of fact, after we filled with human protein, our first choice was actually the endogenous protein from electric eel because it's highly enriched of Navh channels. And but then prior to NavPas, due to our limited experience, we were not able to kind of align the particles. But now with the success of NavPas, we revisited the old data set and we were able to solve the structure of the protein from electric eel, which we named Navh 1.4 because of a sequence homology with a human Navh 1.4 to 4.0 angstrom. And surprisingly, this protein actually came together with an endogenous beta-1 subunit. So sodium channel is also complex with different types of beta subunits, which may help the membrane trafficking or the modulation of channel properties. So this kind of came really as a surprise. All right, now I have the structures of NavPas and the one Navh 1.4 from electric eel. We were able to surprisingly, these 2 structures were in different conformations, which gave us this excellent opportunity to address a number of questions. So first is kind of like the gating mechanism. It really came as a surprise that the intracellular gate of the Navh 1.4 from eel is open, whereas that in NavPas is closed, as you can see. So with this, with these 2 different conformations, we're able to generate a morph to study the, the conformational changes associated with gating. And surprisingly, in addition to this iris-like rotation, you can see each inner helix, S6 helix, also undergoes this axial rotation, which was then identified in a few other, VGIC channels. All right, and another interesting observation. So shown here is a side view of the pore domain only. You can see this hole. This side portal or side hole is called the fenestration. So basically, such side portal is, the one on the next surface. So the one enclosed by the S6 in the repeat 3 and repeat 4 actually constitutes the receptor site, acceptor site for the local anesthetic drugs, okay? And it was shown that these drugs can only bind to the channel when it's activated or inactivated. So our structure shows that, this portal was actually closed in NavPas, but opened in electric eel. So that means, indeed, the side portal is also gated. And this is completely consistent with the electrophysiological analysis, all right? And it provides a perfect interpretation for the state-dependent drug binding. Okay, now, we also have the opportunity to study the ion selection. As I told you, the eukaryotic sodium channel contains DEKA at corresponding locus in each repeat at the so-called SF vestibule, okay, select filter vestibule. And to our, originally we thought, this K, this lysine residue may form, alternate interaction with either D or E, to serve as a gate, right? But to our surprise, this residue is very well resolved in both NavPas and Navh 1.4. It just stays there, pointing, downward into the central cavity. So that means it's kind of static. At first, we thought maybe something was wrong. Maybe it's because of the lack of sodium ions. Anyway, I mean, totally different from the bacterial homolog, as told you. And then, we again collaborated with my colleague, Haipeng Gong in Tsinghua University to perform MD simulation, which actually gave us the opportunity to understand the sodium permeation. As you can see, this lysine residue indeed just stays there very, stably. So basically, the role of this potassium, this lysine and also the alanine, Ka, will just stay there to prevent the ion from going to the other side, to kind of push the ion to go through this, site constituted by 3 acidic residues. So this gave us kind of like the first glimpse of how, a sodium ion penetrates the DEKA site. But this is quite preliminary because we still don't understand how it selects between sodium and calcium and potassium. All right, so the next fascinating question is about the electromechanical coupling. That is how the channel senses the membrane potential, and how is the conformational change of the voltage sensor translated to the gate opening, right? And to cut the long story short, so basically surprising that these two channels, that of NavPas and Navh 1.1, were actually in completely different conformations, even in their VSDs. So based on this, we were also able to reconstitute the morph to illustrate the conformational changes of the voltage sensors, as shown here. This is VSD3. This is the S4 segment that carries the positive charge residues, as you can see now. So basically, these are ion 1, ion 2, and another acidic residue S4, and you can see how these gating charge residues come across, right? Come across this occluding residue to achieve the gate charge transfer, and how this conformational changes is coupled to the, nearby segments in the pore domain and in the linker region. All right, so now, I know I have told you a lot of information. So the take-home message is, previously it was suggested that the linker between S4 and S5 is the key to mediate the so-called electromechanical coupling. But structure comparison of these two channels suggests that, the intricate, the kind of interaction between any adjacent segment in the channel would be, is important for this coupling, okay? And, this information is important because that provides the framework to understand a lot of disease mutations I will show in a minute. Okay, so the last point I'd like to make, I'm sorry, maybe I have gone over time, but this is really exciting because finally we'll have a better understanding of the so-called fast inactivation. So shown here is again, the so-called ball and the chain model to, mediate, the fast inactivation, right? So this model was challenged. And indeed, when we solve the structures of NavPas and the calcium, the closely related CV1.1, to our surprise, we see like, on the 3,4 linker, which carries, this IFM motif, is actually sequestered by the, globular C-terminal domain, okay, as shown here. This is the same in these two structures. So we thought, something must be wrong with the ball and chain model because it cannot be placed, right below this intracellular gate. But then but we just expressed concern. We didn't say it was wrong. Fortunately, we didn't say it was wrong, because after we solved the structure of Navh 1.4 from the electric eel, as you can see, this linker actually undergoes a huge conformational change. And this IFM motif is actually placed indeed close to the channel, but it's not to block this intracellular gate. Okay, this is the intracellular gate. It's actually placed on the other side of the wall. Okay, it's inserted into the corner enclosed by this inner helical bundle and the S4S5 ring in repeat 3 and repeat 4. And this is the morph to illustrate this conformational change. So basically, upon activation of VSD4, which undergoes conformational change, this 3,4 linker is also dragged away to place this IFM motif into the corner outside the intracellular gate. And if you think about it, when you have bundle here, if you insert something, that would push the closure of this inner bundle, isn't it? So with this, this structural observation, we proposed the so-called allosteric blocking model. So basically kind of a modified ball and chain model. So it's not a ball within blocking the gate, but on the other side. So it's allosteric blocking. And something cool about this model is all the residues involved have been characterized in the past 3 decades. So this model actually consists very well with the mutagenesis analysis, which cannot be explained by the direct ball and chain model. So now, this structure really provides a beautiful picture to, reconcile this functional study and mutagenesis analysis. All right, so with that, I'd like to summarize what I told you today. So we solved the structures of 3 eukaryotic related channels, that of NavPas and Navh 1.4, and also the calcium channel from rabbit. And these structures kind of established a framework for mechanistic understanding of ion selection and voltage gating. Speaking of ion selection, so I told you that several different types of channels belong to the VGSE family. Now we have structures of almost each class. So basically, when you examine the molecular basis or chemical basis for ion selection, is really about chemistry, okay? So as you can see, for potassium ions, it's always the carbonyl oxygen, the backbones that coordinate this potassium. And in terms of calcium channels, it's always the carboxylate group, either from aspartate or glutamate that coordinates, coordinates the hydrated calcium ions. And depending on the width and the length of this vestibule, the channel can be, more selective or less selective or even non-selective. And then, for sodium ions, of course, now we have a better idea how DEKA together constitutes the selective filter. All right, and then the structure provides a template to map the over 1,000 disease mutations in the human sodium channels. So as I told you previously, when we map these mutations to the models of the channel, you can see they are almost everywhere, right? There are several hotspots, but basically they are everywhere. And it was really difficult to understand why, some mutations on, some position is kind of pathogenic. But now, as I told you, the structure comparison suggested that, any interactions between adjacent segments is important for electromechanical coupling. Alteration of these residues may change either the dynamics or kinetics of this conformational change, hence leading to the alteration of activation or inactivation. Kinetics is also important for these channels, right? So if the channel is kind of constantly active, that may cause serious problem. So the structures really provide a kind of alternative interpretation to understand these disease mutations. And eventually, the structure provides a template, a better template than the bacterial homolog and potassium channels for drug discovery in the future. So with that, I'd like to thank all the very talented lab members and collaborators. So all this work was done actually in Tsinghua, although now I'm at Princeton University. So basically, Zhou Qiang, I'm just so lucky to attract him into my lab 3 years ago. So without him, I don't think we could publish any of the structure. And Huaizong was the one who identified, very smart, right? Not just brute force effort, but just to screen for the shortest homolog. He was kind of under the effort. He's kind of a leader in the structure determination of NavPas. And Jen solved the structure of the EO protein. A few other members provide instrumental kind of help. So basically, we're very lucky to have, a fabulous EM facility in Tsinghua University, and it's very well maintained by Dr. Jianlin Lei and his team. And also, they give us kind of excellent training on the data collection, data processing. All right, so with that, I'd like to thank you again. Oh yeah, this is just to show that I moved to Princeton last year. This picture was actually taken not by me, but by my colleague. I stole it from his WeChat. Yeah, okay, now thank you again. I'm ready for the questions.
Any questions?
So, actually, in terms of the ion permeability, it's a one-way static or two-way static?
Two-way.
So
it's channel, so it depends on the gradient.
So yeah, then the selection, how does selection work from outside of the membrane and inside the membrane?
So the ions are small. So below the selective filter is a big cavity. So it's the same, whether it's from the intracellular side or extracellular side. It's kind of a two-way process. It's just the same thing.
Any other questions?
Yes?
So can you say a little bit more about the lysine residue over there? Because you said it's only there to prevent the potassium pathway.
Not potassium, the kind of sodium. It seems like in our preliminary MD simulation, it seems like the alanine and the lysine are just staying there to prevent, kind of the arrival of any cation to this site, so that these ions would go to the other. Kind of it's pretty wide, right? But when you have it, they can only go to the other corner to go through, the DE site.
Sure, but why would it need to be a lysine? Because any positively charged amino acid site.
So when you mutate that to arginine, basically its loss of function probably is too big to make the vesicle too narrow to permeate any cation. And then there are only two, right? I mean, histidine is pH dependent. Histidine is kind of has this imidazole ring that may not work well with this site.
Just wondering if you varied the detergents to affect the micelles and if that had any effect on your
detergent, yeah, so in both cases we use digitally, and indeed that's a very good question. So in the structure of Navh 1.4, so the kind of intracellular gate is open. This kind of surprising because we assume it should be kind of closed because it's kind of like a kind of it's a lengthy purification duration. But then because it seems like digitally actually penetrates this intracellular gate just to keep it open. So it's kind of a serendipitous discovery that the channel is open because of the presence of the detergent molecule. Okay. And I would also like to say that something I haven't told you was that we also observed up to 8 lipid molecules between the VSDs and on the pore domain. So that shows that if a lipid, it binds there kind of tightly, that cannot be replaced by detergent.
Since you already started to talk about lipid, do you see anything between the lipid membrane with the voltage sensing fragment from your Cryo-EM densities?
Within the voltage?
Because the voltage sensing fragment probably is charged, right? How they interact with the membrane?
No, these residues are kind of embedded, not embedded, but enclosed within the domain. So it's isolated from the lipid bilayer.
Okay.
Yeah, it's a kind of water-filled cavity. The 4 helices protect these gating elements within.
Okay. Any other questions?
Yeah, question. Can this technique be used to look at, say, for example, lead poisoning, arsenic poisoning, things like that? Shortening the gates and things like that?
The light poisoning?
Lead poisoning. A lead poisoning. Say, for example,
poisoning is more like is related to sodium channel or water channel.
Yeah, but I'm just saying that can this whole technique be used? Say, for example, people have high concentrations of lead in their blood, and so, for example, they could short out these channels. Is that possible?
Well, because there are kind of different types of channels, receptors, right? So if I remember correctly, sodium channels are not the target for that. That mostly affects the consistency in other channels.